What I thought I'd do today was spend about the first 2/3 of my time here to describe to you a story, and it's going to be a short story because of time, on a project we've been pursuing over the last several years, looking at the mechanisms that regulate cell fate decisions and differentiation in a neural stem cell lineage. And then I wanted to spend just a few minutes at the end, maybe 5 or 10 minutes at the end, to tell you about what I've been up to for the last year or so with the BRAIN Initiative, and to give you an idea of where the BRAIN Initiative is heading in this second phase. So as David mentioned, I spent 27 years as a faculty member at UC Berkeley, great time of my life. And the work I'll describe to you, it represents work that was done at UC Berkeley that we are hoping to, well, will be continuing at my lab at NIH. So understanding the regulation of stem cells and how they generate all the cells in the body is a fundamental problem in biology. So as shown in the schematic here, there's a hierarchy of stem cells in development, starting with a totipotent stem cell of the one cell embryo. And then, leading to the pluripotent stem cells, the multicellular embryo, and then again to restricted stem cells that give rise to the cells in the different tissues of the body. Now, as a neurobiologist, of course, I'm interested in the nature of stem cells in the nervous system, in part because there are certain brain regions that conduct active regeneration and activity of these stem cells throughout adult life. Now, the role of adult neurogenesis, I think, is quite interesting. It's important for supporting, we believe, plasticity, processes underlying learning and memory, as well as regeneration and repair following injury and also during aging. Now, there are two very famous sites of active neurogenesis in the adult brain or postnatal brain, the hippocampus, as well as the subventricular zone, which gives rise to progenitors that ultimately migrate to and mature into the granule cell interneurons of the olfactory bulb. Now, there's a third part of the nervous system. Perhaps we don't consider this to be the central nervous system, but also a site of active neurogenesis, and that is the olfactory epithelium in the nose. So we've adopted the olfactory epithelium as a model for studying stem cell fate decisions. There are adult tissue stem cells here that support the replacement of olfactory sensory neurons and other support cells over the life of the postnatal animal. And what's really quite interesting is that if one injures the epithelium and kills off all the mature cell types in that epithelium, these stem cells can fully reconstitute the cell types over the course of a matter of weeks. And so we would like to identify the conserved strategies that are used here for regeneration with the hopes that perhaps it can give us some lessons for clinical translation and ultimately into cell replacement therapies. So here's just a very crude schematic of what the olfactory system looks like in a rodent. Here we have the nasal cavity, the olfactory epithelium. This is the system we study. Of course, these sensory neurons send their axons to the olfactory bulb, which then carries that information up to higher cortical centers. So we'll be concentrating here only on the actual olfactory epithelium itself. Now here's, again, a schematic of the olfactory epithelium. It's a pseudostratified epithelium composed of a relatively small number of cell types. We have the subsentacular or support cells whose cell bodies or nuclei reside at the very most apical part of the epithelium. Mature sensory neurons, their cell bodies lie about 2/3 of the way up. And underneath, closer to the basal lamina, are these so-called basal cells, the globus basal cells and horizontal basal cells. There are cells of the Bowman's gland, and not shown here are another cell type called microvillus cell. Now, it's these basal cells that are documented to be the progenitor cells of this neural stem cell niche. So on the very bottom are these horizontal basal cells. They're flat pancake-shaped cells. These are multipotent cells. Under normal conditions, they're quiescent, but upon injury, they can be activated to reconstitute the entire epithelium, so giving rise to both a neuronal lineage, sus cells, and other cells in the epithelium. The globose basal cells, which lie just above those, these have more restricted potency. They give rise mainly to the neuronal lineage as well as the microvillus cells and a few other cell types. They are constitutively proliferative and also contain transit amplifying cells. So there are a number of questions we would like to address here. One is, do these HPC stem cells enter a transient activated state before moving on to self-renew or differentiate? Another is, where in the lineage are cell fates chosen? We know that these HPCs can give rise to 3, 4, maybe 5 different cell types. Where are those cell fates decisions being made? And are different strategies used to balance self-renewal and differentiation during homeostatic tissue maintenance versus injury-induced repair? And finally, what are the genetic and epigenetic mechanisms governing tissue maintenance and regeneration? So these are the questions we would like to address. Now, I'll just summarize for you very quickly a study that we conducted back in prior to 2017, but published in 2017. This was an effort led by a remarkably talented former postdoc in the lab, Russell Fletcher, with a whole team of students and other associates. And here we use single-cell RNA sequencing combined with clonal lineage tracing under conditions of homeostatic tissue maintenance to characterize the branching trajectory that these stem cells take as they give rise and differentiate to these different mature cell types. So what we found from this analysis is that the first decision point occurs here, giving rise to either to sustentacular cells or these proliferative globospatial cells. And this decision point happens before any cell divisions have taken place. And then these globospatial cells then go on to give rise to the microvilli cells, the cell type I mentioned to you before, as well as to continue dividing as these immediate neuronal progenitors into the population of olfactory sensory neurons. And using conditional gain and loss of function alleles for beta-catenin, we showed that canonical Wnt signaling is both necessary and sufficient to drive these cells toward their neuronal cell fate. So what I'll concentrate for my talk on here is what happens during injury-induced regeneration. So there's a really nice model for studying injury-induced regeneration in this neural stem cell niche. One can kill off all the mature cell types in the epithelium by injecting a protoxin called methimazole that's taken up by the sustentacular cells, which are loaded with cytochrome P450, which converts methimazole to a toxic product, which kills off the sustentacular cells as well as other cells around them as a bystander effect. And what I'm showing you here is our experimental paradigm where we have labeled the horizontal basal cells here and all their descendants using a keratin K5 Cre gene driving Cre-ER, coupled with a ROSA YFP reporter. So we're able to label not only the HBCs, but all the descendants that come from them. If we look in an uninjured epithelium here, you can see these, the YFP reporter is localized just to this monolayer of horizontal basal cells that are quiescent along the basal lamina. If we treat these mice with methimazole and look at them, the epithelium at different time points, at 24 hours, you can see a little bit of something happening here. By 48 hours, you can now see a bilayer of cells that represents cells that have come about after the first set of cell divisions. The mature part of the epithelium is now sloughed off. These cells have died off and gone away. By 7 days, we're seeing the appearance of sustentacular cells. Globospatial cells are already apparent by 48 and 96 hours. We're starting to see the appearance of immature neurons. By 14 days, we can pretty much see all the mature cell types in the epithelium. And by 4 weeks or so, the neurons have already innervated the olfactory bulb. So this is our experimental paradigm. What we've done is we've performed single cell sequencing at different time points in similar studies on these lineage trace cells and watch to see what cells come about and what are their properties as they move through these different transitions. So on the left is just a T-SNE plot. Cells are color-coded according to cluster analysis that was done independently. I've given you a little cheat sheet on the right. Let me just draw your attention to what these cells do, what stages they progress through over these different time points. So I'll show you this here on the bottom. Again, we're starting out in the uninjured epithelium. Here are the resting horizontal basal cells, again, normally quiescent. And what was really quite remarkable to us is that within 24 hours, all these cells shifted en masse to what we're calling this first activated state. So they just, boom, move right over. A second activated state, HBC2. And what characterizes these cells that's quite different from the early transition states from homeostatic differentiation is the appearance of a set of canonical wound response genes that are typical in other wounded epithelial structures, as well as the expression of cell cycle genes. Now, by 96 hours, we're seeing the appearance of sustentacular cells here, as well as renewed HBCs. So as any self-respecting stem cell will do, it has to not only differentiate, but also give rise back to the original cell type. And then by 7 and 14 days, we're seeing progression through the neuronal lineage. So we really want to concentrate on what's happening during these early stages of this stem cell activation process. Okay, so what was really interesting to us is that in spite of these robust changes in gene expression, which can be seen by this large shift in T-SNE space, but also in these volcano plots showing the up and down regulation of a large number of genes, we observed a relatively small number of transcription factors that showed similarly robust changes. And on the bottom here, I'm showing you these so-called graph or hairball plots. Hairball plots, I think it's pretty obvious why it's so named. The nodes of these graphs represent transcription factors that show correlated expression with other transcription factors. And I've color-coded them here based on the clusters in which they're most highly expressed. Now, the activated HBCs here in light blue, you can see they're not represented in these graphs. So it really begged the question of how is this very rapid shift in gene expression accomplished that is both so robust and so rapid. So we have a hypothesis that the genes upregulated during differentiation are poised for expression in the quiescent horizontal basal cells. And to investigate this further, we use a technique called ATAC-seq, which is assaying for areas of open chromatin and therefore genes that are either poised to be expressed or are being actively expressed in the cells of interest. So these studies were carried out by former postdoc Tiffany Cho. Let me just walk you through what these assays look like. So these are now bulk analyses. We're looking at whole tissue, either on uninjured or injured tissue. And on the left, I'm just showing you as an example where we've grouped genes that are either highly expressed or silent, in this case, in the uninjured HBCs. Each row corresponds to a gene, and the dark color red on this heat map shows regions along the chromosome corresponding to the transcriptional start site that are either in an open conformation or accessible to this assay, to the transposase, or inaccessible corresponding to these silent genes. So that's what you see under what you'd expect to see typically. And let's look at some example genes just for show. So here I'm just showing you two genes that are expressed in resting HBCs. And here are 4 genes on the right that are upregulated in these activated HBCs. Now, this is all a bit too much. Let me just draw your attention to 2 examples. So on the bottom 2 traces, this shows RNA sequencing data from either resting or activated HBCs. In this particular case, it's ICAM1. It's the cell adhesion molecule that keeps these cells stuck down to the basal lamina. And you can see high expression, lots of traces over the body of the gene in resting HBCs, and it begins to be downregulated upon activation. And here we can see chromatin accessibility over the transcriptional start site of this ICAM1 gene in the resting state, and some accessibility, but diminishing in the activated state. So just what you might expect for a typical gene. Now, what we see is uneven, but here is SPAR1, which is one of these canonical epithelial wound response genes. It is not expressed in resting cells, quickly upregulated in activated cells. And here, quite interestingly, you can see that around its transcriptional start site, it resides at open chromatin in both the resting as well as in the activated state. So we found this to be quite interesting. And we asked now, what could we learn by extending this analysis to the single cell level? So Tiffany continued this analysis together with Koen Vandenberg, a terrific statistician we've been working with. And we've plotted cells analyzed by single cell ATAC-seq, again, looking for regions of open chromatin in these cells at the single cell level. And we've combined here in these UMAP plots, both cells coming from injured as well as 24-hour, from uninjured as well as 24-hour injured epithelia. And I think what you can see here are 3 main neighborhoods with an overlap between the uninjured and injured conditions. And we've ascribed to these different cell states. The injured corresponds to resting. The uninjured corresponds to resting. The injured corresponds to activated. And we see this overlapping cluster, which we call the hybrid cluster, which indeed represents cells that arise from both uninjured as well as injured treatment. Now, if we dig a little bit deeper and look at if we now can look at the accessibility of chromatin corresponding to transcription factor binding sites, transcription factor binding motifs in the resting and activated cells actually are consistent with the experimental origin of these cells. And quite interestingly, in the hybrid clusters, we see an enrichment of genes regulated by multiple FORCAD transcription factor family members. So it really starts speaking toward the coordinated regulation of genes manifested at the chromatin level, the epigenetic level. So this gives rise to the question of whether this hybrid cluster represents cells that are poised for activation before that process is actually played out at the gene expression or RNA level. So let's come back to the RNA expression data that I showed you before, where the phenotypes of the readouts of the different cells are manifested overtly. Now, from the early experiments we did using SmartSeq-based technology, we just didn't have that many cells to know for sure whether there was in fact a so-called hybrid cell type or cell stage that manifests at the gene expression level. So we went back and did more experiments using droplet-based methods. Now we have this nice lineage analysis using over 21,000 cells. I've extracted out here for you just the resting HBCs and just the activated HBCs that were derived from tissue 24 hours following injury. So this is what it looks like. So we still don't see, even with a much greater number of cells analyzed, we still don't see this hybrid cell type in the RNA sequencing. So next what we did is we used a statistical algorithm to predict which genes might be expressed or differentially expressed in the hybrid cluster based on chromatin accessibility. So in other words, we now look at the ATAC-seq data. We make a prediction of what genes should be expressed based on their openness. And then we see where those genes are actually expressed in the RNA sequencing data. And what we see is that genes that are predicted to be expressed from the ATAC-seq data are actually expressed and activated or resting. And the ones that are predicted to be expressed in this hybrid cluster are actually expressed in the activated cells by RNA sequencing. So this suggests that these uninjured cells in the hybrid cluster may represent a latent activated state that's poised for regeneration. So there's clearly a lot more work to be done. But I think what I've shown, I hope you believe what I've shown you so far, is that injury elicits a rapid shift from resting to activated stem cell states. And a genome-wide analysis of chromatin accessibility reveals a latent activated state that's not detected by RNA sequencing. And I think what I've shown you here is some evidence that a subset of quiescent stem cells are poised for activation. And this is something that we've seen in embryonic stem cells. And more recently, there's been a beautiful study that's been posted in bioRxiv from Sergio Pasca and Will Greenleaf, showing that cortical stem cells that are destined to become astrocytes actually show a similar kind of pre-activation in the progenitors prior to the differentiation of those cells. So I think this is going to be a general theme in adult stem cells and certainly in neural stem cells. And the open chromatin corresponding to binding sites for related transcription factor families suggests a coordinated epigenetic regulation of stem cell pre-activation. And we have an ongoing interest in dissecting these mechanisms further, including looking for the signals that initiate stem cell activation, as well as the downstream targets and effectors that are carrying these programs out. And just as a shameless plug for the lab, we are now hiring and looking for great, talented postdocs to continue this work. Okay, and I must thank the people that did the work. Again, Russell Fletcher in the lab led the early efforts. Tiffany Joe took over and conducted the ATAC-seq experiments, and all the work we've been doing over the past, actually literally 20 years has been done in close collaboration with my terrific statistical colleagues at Berkeley, Sandrine Dudois and Elizabeth Purdom. We've had very generous funding from various sources, and I just want to acknowledge the folks that really made all this work happen. So let me now transition to what's been my day job for the last year or so, and just give you a very quick overview of the NIH Brain Initiative, the so-called Brain 2.0, or the next phase of this very exciting initiative. Now, the mission of the Brain Research through Advancing Innovative Neurotechnology is a brain initiative. It's to revolutionize our understanding of the human brain by accelerating the development and application of innovative technologies. This was announced by the White House in 2013, and with the first awards made in 2014. It represents a partnership between 5 US federal agencies and many private foundations as partners. The initiative has been guided by 2 strategic plans that were commissioned by Dr. Francis Collins, director of the NIH, One is the Brain 2025 report that was issued in 2014, followed by the Brain 2.0 reports that were issued in late 2019. David Anderson played an instrumental role on both of these, and I think some of the other speakers here have as well. So the goal of the NIH Brain Initiative is to develop and apply new tools for understanding how neural circuits underlie complex behaviors, both in health and disease. I think to accomplish these goals, we need to leverage the technical innovations to enable new discoveries about neural circuit function. We then need to use these discoveries as a foundation for new therapeutic strategies for human brain disorders. And very importantly, we need to disseminate and democratize these technologies, both for basic discoveries as well as for clinical applications. Now, I just want to go over for you where we are in terms of the phasing of this project. And here's what the funding outlook looks like for now. We're right now in fiscal year 2021. Again, the initiative was started in 2014 with a very modest, by today's standards, investment on the part of NIH of $46 million. The funding has grown by quite a bit. I just note for you that funding for the Brain Initiative at the NIH comes from two main sources. One is the so-called base funding, which goes to the 10 participating institutes and centers at NIH. And then on top of that, in this lighter blue, teal blue, is funding from the 21st Century Cures Act. Now, the Cures Act funding runs through 2026, which gives us a nice increase over the base funding. And we are very fortunate this year to receive an additional $100 million in funding from Congress, which brings us to a current level of funding of $560 million a year. Now, those of you in the audience will notice that there's a large bump up in funding from the Cures Act funds in 2023. And with encouragement from the Brain 2.0 report, we're looking toward this big boost in funding in 2023 to launch large projects that have the potential to transform neuroscience research and the impact these might have on human health. So I just want to take a minute to describe to you what these large transformative projects are. We've chosen three of them. The first is a project to organize resources for brain cell type access and manipulation across species, the so-called cell type specific armamentarium. This is really to develop ways of getting into cells, monitoring them and modulating them in multiple mammalian species, including big brains, non-human primate brains, with the ultimate goal of supporting new therapeutic strategies for human brain disorders. The first RFA for the pilot studies has been issued, with receipt dates coming just this next month, as well as in October 2021. The second one is the phase 3 of the brain cell census project, the so-called PARTS. This will build on the remarkable success of the BICCN mouse brain cell census that Honghui Zhang so beautifully showed you earlier this morning. And the idea here is to shift the emphasis to construct a comprehensive human brain cell atlas. And I should note that in our fiscal year 21 appropriations, a significant amount of funds were earmarked to support the acceleration of the launch of these two projects. The third project is the so-called, something bad happened.
Are you not able to advance, John?
There we go, is the so-called next generation technologies for brain marker connectivity analysis, or the wiring diagram. And here the goal is to decide whether and how to obtain high resolution, comprehensive connectomics maps of entire mammalian brains. And just as a heads up, we have a series of 5 workshops coming this spring that will really ask the hard and deep questions to help guide how this project will go. So, again, one of the main goals of the NIH is to devise new cures for human disease. And I think it's fair to say that discoveries supported by the Brain Initiative will lay the foundation for some novel human brain interventions in human brain disorders. Brain-supported technologies are unraveling the circuits and computations underlying complex behaviors. And this will enable the identification of cells and circuits affected in neurologic and neuropsychiatric disorders. We're already making tremendous progress, not only with brain funded projects, but across the board. We're already seeing more and more sophisticated closed loop deep brain stimulation devices for conditions such as epilepsy and movement disorders. We've already heard here in this symposium from John Donahue some great work that's being done to develop sensory and motor neural prostheses. And I think the blue sky potential of some of the technologies and tools that we'll develop, in part based on the cell census work and the cell access work, will be targeted molecular gene therapies for these human brain disorders. And so as we move deeper and closer to the human condition, it's very important that we integrate ethical considerations in all that we do. And to this end, we take this very seriously. Brain has a neuroethics working group who's working very hard to make sure not only that we're doing the right thing with current projects, but to look ahead to make sure that we continue doing the right thing. We've established a set of neuroethics guiding principles that, because we're dealing with the human brain and the human mind, in addition to critical issues surrounding privacy and safety, there are also very important issues surrounding agency, the potential for malign use, dual use, making sure we engage the public in all that we do, and also ensuring that the riches that we create here are available for the benefit of all. So this is an ongoing process. The other things we're interested and very committed to are training the next generation of scientific leaders. We have a number of training programs within Brain. There's a F32 program. And again, these programs are designed for trainees and future leaders who are interested in pursuing research and education in areas that are supported by Brain. We also have a diversity K99R00 program to enhance diversity in our workforce. And in this case, I'm embarrassed to say that women are considered an underrepresented minority. So this program is open to women and other underrepresented groups. In our sister Blueprint program, there's a similar program to the K99, but one step earlier for late stage graduate students transitioning to postdoctoral fellowships. Again, a diversity program. We're just trying to get more people, more talent, more diverse talent into the game. And very importantly, we have diversity supplements to Active Brain Awards. You can get more information by going to our website or talking to any of our very dedicated program officers. And finally, our goal is to build community. And I just want to give a shout out about our upcoming Brain Initiative Investigators meeting. It's going to be virtual this year. We have high hopes for 2022. Last year, I think we still had a terrific meeting. I encourage you to visit the meeting website to register. We had, I think, over 4,000 people register last year. So we're really able to draw a big crowd. And I can promise you a very exciting program this coming June. And with that, I just want to finish by thanking the tremendous support that I have, that we all have in these efforts as we pursue these projects. There are over 100 subject matter experts that are squarely behind the mission of Brain, and I encourage you to contact me or any one of these folks. You can find them on the NIH Brain website with any questions you might have, and I do encourage you all to get engaged.